Since we're talking about lipids and important lipids in our body, who can tell me those three membrane lipids, those three categories that we talked about? The first one's easy. Phospholipids, that's the first one. Glycolipids, and then the third one that we usually talk about being bad. Cholesterol for us, and do you remember the category we put cholesterol in? Sterols. All right. Well, we also have, as a part of our phospholipids and glycolipids, our fatty acid chains, but we have these things that are called essential fatty acids, EFAs, omega-3s, omega-6s. Where have you ever heard of omega fatty acids or essential fatty acids before? Fish oils. Fish oils. They are to help you with the amount of fats and cholesterols you have in your diet. And specifically these essential ones are important in the membrane structures of your central nervous system or in your reproductive organs. Because central nervous system's regulating a lot in your body, just like your hormones do. But when you look at the purpose, your purpose for being here, your purpose for living, is one thing and one thing only. No, reproduce. Make something that looks pretty much like you and let them carry on the genetic material. Reproduction. So it's not a surprise that we have these essential fatty acids in those membranes. Now, turning back to membranes, membrane fluidity and structure. We talked last time at the end of our lecture. When you have a membrane that melts at a higher temperature than another, what two things are going to attribute that membrane for possibly the reason it has a higher melting temperature? The density, and there are two things that are going to affect that density. The bonds. And so if we have a higher melting temperature, what can we deduce about those bonds and those fats? More single bonds, and so they're more saturated fatty acids. But it may not necessarily be the more saturated fatty acids. What could also cause membranes to have a higher melting temperature? Longer chains, more carbons. Now, the more double bonds, the more unsaturated and polyunsaturated, that's going to lower our membrane melting temperature. But we can also pack cholesterol. See, in our diagram here, we have the little red ring structures. And you see how it's moved the phospholipids further apart, less dense. And at first, you may deduce that, less dense, it's going to have a lower melting temperature. But because of cholesterol and its structure, we're going to see that cholesterol moderates the melting temperature or buffers the melting temperature. It's not simply just going to lower it. So with our cholesterol, as we get above the melting temperature of the membrane, it's going to tend to extend that semi-solid phase a little bit further. It's going to be less fluid than it normally would be above the melting temperature. And likewise, when we look below the melting temperature, it's not going to become as solid as quickly. So do you see, it has an effect on both ends. Instead of a nice little peak to our curve, imagine when you add cholesterol, we have a nice plateau that extends that range on both sides of the melting temperature. So do you see why I use that term buffer? It has an effect on both, and it moderates either the fluidity or the solid phase of a particular membrane. So cholesterol is good. We have to have cholesterol, and it's important in our body. So, as we wrap up and get closer to finishing our discussion of membranes, just a reminder, when we look at all of the proteins that are within our membrane, someone somewhere had to visualize this, theorize this, and put it together. And what scientists have used is something called freeze fracture. You freeze tissue and you freeze cells, and if you fracture, and you kind of have to get lucky to do this, But you can fracture right through the hydrophobic layer of our membrane and split it in half. And when you do, you can look at the outer portion of our lipid bilayer and the inner portion of our lipid bilayer. And with an electron microscope, you can see these little bumps. Each one of those bumps is a protein. And so in using freeze fracture and using, like we saw before, the laser photo bleaching, we have quite a bit of knowledge and understanding about the diversity of the proteins within our plasma membrane. Diversity of structure, and with different structures, we can have different functions. So here's just, again, a short list. We had a much longer list before. Those functions haven't changed. Adhesion, receptors, transporters. We even have proteins on the surface that are going to say, hey, I'm supposed to be here. I'm part of this person. Versus foreign material that will lack that signal. So with our proteins on the surface, there are a number of ways that proteins can interact with the phospholipid layer. One way is we can have what are referred to as integral membrane proteins. Now, many of these are going to go completely through the phospholipid bilayer so that you have an extracellular portion and an intracellular portion, and then a part that goes straight through the middle. These are integral membrane, but in fact, these can also be called transmembrane. Trans meaning passing through. Now, some of the integral membranes aren't actually going to pass completely through. They just may interact with one portion of the membrane. And in which case, if we look at those that are only bound to one surface, we can call these peripheral membrane proteins. Some of these may interact with the phosphate head, the glycerol head, or the polar group. Some may interact with sugars on the surface with our glycolipids. And then some may actually insert themselves in via fatty acid chains, in which case, if they're using those fatty acids, we say they are lipid anchored. But do you see the difference between our transmembrane, integral membranes versus our peripheral proteins? But regardless, we have portions that are on the outer surface toward the extracellular part and on the inside surface toward the cytoplasmic part. And many of these are going to function what we're going to see in the next two chapters about signaling from the outside to the inside of the cell and vice versa. All right. So with our integral proteins, as we see the pass through the membrane, many of these, not all, many of these are going to have a structure that is an alpha helix. Now, for protein structure, what level of structure is an alpha helix? Secondary structure, sort of facilitated and mediated by what kind of bonds? Hydrogen bonds spin it together in that secondary structure. So we can have one pass, two pass. You may have 7 or 11 passes through, depending on how big and what the function of a particular protein is. But where you see an alpha helix, if it's a transmembrane protein, what do you think the characteristics of those amino acids are going to be that go right through our fatty acid layer? What types of amino acids, not names, but what types of amino acids are going to be there? Hydrophobic. You're not going to put a polar or charged, a whole bunch of those into an alpha helix that's passing through that layer. Those are going to be hydrophobic amino acids. And that's one way that we can really distinguish if we just look at a protein sequence, the primary sequence. Am I looking at the sequence of a membrane protein or a non-membrane protein? If you see all those hydrophobics, it's a good clue that you are. And that's just a higher look at sort of a sequence. Why are there a bunch of glycines? Glycine's not really hydrophobic, is it? It's not polar, so even though it's just a hydrogen, remember that's our honorary hydrophobic amino acid, and it's small. It doesn't get in the way. Now, for our transmembrane reason, we can also use the secondary structure of a beta sheet. Now, that might not conceptually be easy to see. I can see putting a cylinder through the middle, but a beta sheet having a lot of hydrophobic amino acids may not be as intuitive. But now if you look at this illustration, you take that beta sheet and you turn it into what they refer to as a beta barrel. You see what they did? They took a sheet and just rolled it up. I like to do this with a tortilla, especially when you put stuff in the middle of the tortilla. So we'll call this our beta tortilla as we're putting it through the membrane. And you can see how you could create an internal environment that may be very different to the external environment. But again, same sort of thing happening. Hydrophobic amino acids are going to be used to pass through, creating this internal transport tunnel that's going to be used in a lot of our organisms. Now, we can find these referred to as porins. In our body and our kidneys, we have these porins that are called aquaporins. Guess what goes through those? Water. And we see that bacteria and mitochondria have a lot of these beta barrels and the porins. But of course, we also have barrel-shaped channels that are, in fact, instead of having a beta sheet, they're just a bunch of alpha helices organized into a tunnel. So, big point here, hydrophobic amino acids will form the transmembrane part of our integral membrane proteins, whether we put them in the form of a helix or in the form of a sheet and a barrel. So peripheral proteins, in this illustration, the peripheral protein is the darker color. What would the lighter colored one be? Transmembrane. Now, in this particular illustration, the little blue lines, they're trying to exhibit the interaction with lipids, the phospholipid layer. But in addition, you can hang on to the membrane with those lipids, but many of our proteins are going to interact with each other, and this may be, like disulfide bonds holding each other together. And if you have two proteins held together with disulfide bonds, what level of structure is that for proteins? Two proteins held together. See, that's the key for quaternary. Quaternary. Two proteins held together. Anytime you hear two proteins, you're thinking quaternary. Now, much of our interaction out here is potentially also going to be hydrogen bonds. But I think you can see how there's a lot of different flavor and variety of how proteins can hang on just to the surface without spanning all the way through the membrane. Now, another important component of our plasma membranes, whether they're integral or peripheral, is on the external surface, we're going to have proteins that have a lot of carbohydrates on them. These proteins have been glycosylated. And if we're looking for an organelle that principally functions in the glycosylation of proteins, which organelle are we looking at? Okay, the ribosome produces, that's our protein synthesis, what? Protein synthesis is translation. And the ribosomes on the ER are responsible for that, and the beginning, just the beginning part of glycosylation. But the Golgi is principally the organelle responsible for the glycosylation we see, especially on the cell surface. And we have a ton. In fact, when scientists first started looking at cells and they recognized the material on the surface, they recognized the sugar before they recognized anything else and named it the glycocalyx, or the sugar coat that's around the cell. And as diverse as the proteins are, we have tons of carbohydrates expressed on the surface of those proteins. Now you're going to find much more carbohydrate and glycoproteins, these are the proteins that are glycosylated, on the extracellular surface than you necessarily will on the intracellular surface. Because there's a lot of interaction facilitated outside the cell via carbohydrate. So it's not just the protein that's important. So when we look at glycoprotein, certainly we can look at adhesion, for instance. That's a big one. When I worked at the Cancer Center in Arkansas doing studies of plasma cell leukemias, we were working with a molecule that had 3 very long carbohydrate chains. The molecule was called syndecan. And we found in these cancer cells, if they stopped making syndecan, they were very aggressive. They could invade into experimental media very quickly. And when placed into mice, they could form tumors and kill the mice in a matter of a few weeks. However, when we cause these cancer cells to start making this molecule again with those sugar chains, we could put the cells in the mice, and the mice after 6 months showed no signs of anything. We would place the cells in experimental culture. They would not migrate and invade. Well, part of my job was to monkey around with the genes, so I made the genes so that you could make the protein, but you couldn't attach the sugars. What do you think happened when we put those cancer cells into the mice that could produce the protein, but simply couldn't produce the sugars? It was as if the thing wasn't even there. It killed the mice quickly, and experimental media, the cells invaded rapidly. So in that sense, the sugars were the critical part for the function of that protein as it related to adhesion. But embryonic development, carbohydrates, absolutely critical. Defense, being able to recognize your own teammates versus foreign materials, that's going to be important. And then cellular uptake, that endocytosis. Just a few of the functions that carbohydrates on the cell surface are critical for facilitating. Now, here's a last little cool experiment of how we look at protein mobility. We saw the phospholipids with the laser zapping of the fluorescently labeled. Well, in this case, you can make a cell that's called a hybridoma. You essentially take mouse cells and human cells, and when you fuse them together, you can cause these cells to begin to make a lot of antibodies, which is a real useful way of getting reagents to use in a lab. But in this case, they decided to do these hybridomas to investigate, okay, what's going to happen to the proteins that we label? If we label the human proteins in red and the mouse proteins in green, and we form this cell, are we going to end up with a basketball that's green on one side and red on the other side? And what they've discovered, in fact, is over time, and this is only 40 minutes after the cells fuse together, all the proteins have mixed. So this really clearly illustrates the fluid mosaic model and the membrane fluidity, not just the fluidity of phospholipids that we saw in the photobleaching experiment, but now using these differently labeled proteins, we know proteins float around as well. And in 40 minutes to be completely diverse and diffuse over the cell, that's really rapid.
Is that how venom works?
How what?
Venom?
I don't know the specifics of the toxin that's in venom, but venom does diffuse into the cells. And it just wreaks havoc on the cell, and you start getting necrosis just really quickly. You're going to feel really funny, but when you look at the area where you've had the snake bite, yeah, you're going to have big problems, depending on the type of venom too, and the amount. And by the way, I just saw an article, if you kill a snake and you chop the head off the snake, you better not mess with the head, because reflexively, the mouth is still going to be working, and the snake no longer has any control about how much venom is injected. So if you get bitten by a severed head, you're going to get a full dose of venom. So,
Don't grab the back of the head and pull it off.
just best news is you see a snake, avoid the snake. Snake's not messing with you, hurting you, leave the snake alone. Here's a summary slide for your study. Again, we have terms in here like cerebrosides, gangliosides. If we didn't talk about them, don't worry about them. But this sort of gives you an idea of our lipids, fatty acids. We talked about triglycerides. There's our sphingolipids. Remember, that's for humans. And then we've got our steroids over here. Here's our glycerol-based phospholipids. And it really gets down into the very specifics that we saw on some of our charts. But again, I'm more interested in knowing these broader categories. And then this just gives an example of a couple of different types of fatty acids. So here we have palmitate, and here we have stearate. Which one has, if you make a membrane of one and a membrane of the other, of those two, which is going to have the higher melting temperature, and why? What's the first thing you notice about both of them? They're straight, so that means they are what kind of fatty acid? Saturated fatty acid. So if they're both straight, are they going to have a different melting temperature? Okay, for those people that might be ocularly challenged, since I see somebody getting out glasses, this has a carbon number of 16 carbons in the chain. This has 18 carbons in the chain, which has a higher melting temperature. The 18. Because you've got more carbons in there to create a higher density, you're going to need more heat energy to melt it. Here we have oleate and linolate. These are some of the common ones that you have. We have oleate, unsaturated fat. We've got one double bond. Linolate, we got two. We'll still call that saturated. I think you have to have more than two before you're poly unsaturated. So again, just an idea of what the structure looks like in the space filling model, and you can see the bends and the kinks in those unsaturated fats that decrease the density of our membranes.